let's move to the next speaker in the session, which is Professor Gisbert Schneider. He is one of the highest cited chem informaticians and specialists in computational medicinal chemistry. He has spearheaded the fusion of machine learning and AI with medicinal and bioorganic chemistry. He is credited with coining the terms that we'll use, such as scaffold hopping and frequent hitter, not just credited with terms, but has done a lot of research in these areas. He had developed groundbreaking algorithms for de novo molecular design, which have led to profound transformation in the way we attempt to discover new drugs. Gisbert currently is a full professor for computer-assisted design at ETH Zurich. He is also a CEO of Xenogis LLC, bridging the gap between academia and industry. He is recipient of multiple awards. To highlight a few, it's Ernst Sharon Prize, Herman Skolnick Award from the ACS, and a Mellon Beilstein Medal. Gisbert, your talk is on de novo design. Please.
Thank you very much, Alex. It's a pleasure discussing some of our work and our ideas revolving around de novo design. So we're actually combining machine learning and applied medicinal chemistry under one roof in one lab. So we make new molecules and challenge AI tools and machine learning tools until they break to see to which degree can we use a certain technology for de novo design. Now, why is it so difficult to come up with new drugs? One reason is that the cardinality of the search space is virtually infinite. So the task is to pick one molecule or 10 molecules, maybe 100 molecules from a virtually infinite search space. Traditionally, this has been done, and it's still one of the driving forces of drug discovery is high throughput screening, HTS, but also virtual screening, as we already have heard in the preceding talks, can deliver useful compounds which we can test in the lab. Now, de novo design, and in particular, a particular aspect of generative AI, which I will highlight throughout my talk, inverts this concept. We're no longer screening for new molecules, but we're generating the molecules we want to see. So in a way, we can avoid this problem of the high cardinality, the vast search space, by asking a computer algorithm to come up with hopefully optimal or at least locally optimal solutions in this molecular space. So de novo design reshapes the approach to drug discovery by inverting the perspective from screening to designing molecules. It has the potential to complement and potentially replace high throughput screening, at least to some degree. The most important aspect of de novo design, I think, is that these tools come up with surprising novel ideas which we haven't considered or a chemist hasn't considered at the time. So the aha effect caused by de novo design algorithms is, in my view, one of the most important assets we have when we use these tools. Before I dive into the world of de novo design algorithms, I'd like to mention that we're working at the edge of chaos when we interfere with living biological organisms. We're facing the problem of error, of models, of data. We're facing the problem of non-linearity of drug action, non-additivity. We heard about activity cliffs in the previous talk. And this all boils down to partial predictability. We have to concede our imperfect understanding, an incomplete understanding of the molecular pathology and the molecular causes and mechanisms of disease. Given that, and accepting the fact that we will not be able to make perfect predictions, also as highlighted by Marty earlier on this morning, then we can, of course, use de novo design with AI in a very fruitful manner. And because drug discovery is not a classic engineering discipline, we have to cycle through our discoveries, and the molecular design cycle is highlighted here in this cartoon. We start with a hypothesis, and the hypothesis in chemistry is a molecular structure, actually, which we then synthesize in the lab. These molecules have to be tested and validated, and then based on these results, we can learn again and refine our hypothesis. Over the past years, we've seen a clear move from human-generated hypotheses to machine-generated hypotheses and learning. And at the same time, robots take over certain parts in the laboratory. So the idea, the concept is already out there to have fully automated this drug discovery labs. And we will hear about some of these today and tomorrow. Chemistry, and medicinal chemistry in particular, lags behind when it comes to automated design. Here is an example from architecture. On the left-hand side, you see a generative model of such a hallway with columns. And on the right-hand side, you see a concrete printer, which has been built at ETH Zurich by my colleague Benjamin Dillenburger. And once you have the design, these 3D printers immediately generate the building blocks for new architectural designs. If it were only so easy with chemical synthesis, and I will show you a few examples what works and what doesn't in chemistry, and how AI could potentially help us overcome this bottleneck of chemical synthesis in the discovery cycle. Now, for de novo design, we need to solve three general tasks. One is structure generation, the second is scoring and selection, and the third one is actual optimization of the hypothesis. There are various tools available to generate new structures. Scoring and selection is actually the most critical one, and here we have the greatest need for novel ideas and prospective research. And finally, there are in particular certain active learning algorithms available for optimization. Let's start with method one. I will present to you 4 de novo design methods, and I start with the historic one because it's still quite actual and it's quite useful. Namely, de novo design in a very low data scenario when there is maybe just one compound available which you wish to mimic and make a scaffold top. The idea is to collect small molecular building blocks and annotate these building blocks in a database as to which potential chemical transformation they could be used in. A rule-based architecture underlies is the basis here of this machine learning tool. So you define building blocks and chemical transformations, and then you can enumerate and iterate through these virtual chemical spaces. These approaches, rule-based molecular design, are extremely valuable when only limited data is available. And at the same time, with a new design, because we use building blocks and chemical transformations for constructing these virtual molecules, they come up with a potential synthetic pathway for each generated molecule. So they can be fed directly into an automated synthesizer to produce the molecules generated with this machine learning approach. I added this particular slide because earlier on the question arose, what's the difference between scaffold hopping and de novo design? And here you see the very first application of ligand-based de novo design with a machine learning de novo structure generator and a fully automated optimization algorithm, starting from a template molecule, a single molecule as reference, which you wish to mimic. Then the de novo design tool comes up with a suggestion that needs to be synthesized in the lab and tested. And this result is then fed back into the learning algorithm for iterative combinatorial optimization. And in fact, when we apply de novo design in such a fashion, then de novo design is an instance of scaffold hopping. Here is another example how one can use de novo design and these rule-based de novo design tools. For example, when you wish to hop away from a complex natural product scaffold. For example, here you see anglerine A. It's an anti-cancer sesquiterpene, which has been de-orphaned by target and activity prediction as a menthol channel, calcium channel, TRPM8 blocker. But it requires a total synthesis of overall 11 linear steps. Now, de novo design has come up with two novel chemical structures. These could all be synthesized in 3 steps as predicted by the algorithm and have the same activity as the original template. So again, de novo design can help us generate new structures. These are IP-free, so one could patent those structures. They can be synthesized and they mimic, in this case, a complex natural product template. Here's a more recent example of the application of rule-based de novo design. Another natural product, marinopyrrole A, was converted into this de novo structure, and this de novo molecule actually is the most selective COX-1 inhibitor we know to date. And I'm showing this particular example because when we look at the de novo generated molecular scaffold, we see that it is quite similar, not identical, but similar to celecoxib, a known COX inhibitor. This instance of de novo design showcases the ability of these tools to generate chemical structures that are known in the domain already, but without ever having seen these molecules. In order to find out why this particular de novo design does not act like celecoxib, but is a selective COX-1 inhibitor in contrast to celecoxib, of course, then experimental structure determination is of the essence to have hard facts to pursue hit-to-lead optimization. So here is a link between ligand-based de novo design and structure-based follow-up. Method 2. We're moving on in history. More recently, chemical language models have become fashionable, and we too had a close look at chemical language models to learn the syntax of drug-like molecules from their small string or selfies string representation. So the first step of chemical language modeling is to define a training set of training data that is suitable for the particular task, represent these molecules as, in this case, small strings, and then in an autoregressive fashion have the CLM tool understand, if I may say so, the syntax of small strings so it can reproduce the input molecules of the training data. Once done, we can use such a tool to generate new molecules based on the internal probability distributions learned. Now, all these tools, these deep learning tools require data to learn from. We typically rely on chemical databases, supplier catalogs, patents, or as we just heard from Alexander Warneck, virtual compound libraries, for example. Compared to other areas of deep learning, for example, face recognition, computational drug design suffers from scarce data. Therefore, it is essential that we define context-specific data selections for a task at hand and represent the molecules in a suitable fashion. And here, human ingenuity, human knowledge, chemical knowledge plays an essential part. We cannot expect a deep learning tool, for example, a chemical language model, to learn everything there is about chemistry and about the relevant molecular properties just by feeding in more and more data. This is an observation and experience we've made over the past years working with these tools. Now, a natural language to learn for such a chemical language model is the amino acid single letter code, for example. Here is an example of an automated design, make, test pipeline where we trained a chemical language model on alpha helical sequences. We wanted to generate, ask the computer to generate new anti-cancer peptides. So the system was trained with known alpha helical sequences and known membranolytic peptides. Synthesized on a peptide synthesizer, and then the molecules were tested here, as you see in this video, on cancer cells. It's what you see here is a breast cancer cell, an MCF7 cell that contains a fluorescent dye, and when one of these de novo generated peptides is applied here, then the membrane is burst and the cell leaks and dies. And the most effective peptide here turned out to be a cancer cell selective peptide that leaves healthy cells intact. When we talk about full automation and integrating the design, make, test parts of such an automaton, we can build all kinds of machines. And here's an early prototype we've built that fits on a desktop, a benchtop, in a way portable de novo design laboratory, and that was guided by a chemical language model, which we trained on vendor catalogs. So on synthesizable molecules, because we wanted the language model to learn something about synthetic accessibility of the designs. And overall, in this particular application, we had more than 50% successful syntheses, and again, more than 50% of those molecules synthesized had the desired biological activity. And that is a typical outcome for de novo design with chemical language models as well as with rule-based de novo design tools. One can expect at least a 50%, it's a conservative estimate, a 50% chance of being synthesizable and bioactive. Now, when we look at these molecules that are suggested by a chemical language model, we see that approximately 50% of these suggested designs are actually new, and the others are known in one or the other way. And I think this is a healthy mix, because we don't want to create just any arbitrary new molecule, but we want to see these new molecules as similar to what is known, because this increases the chance that they are synthesizable and have a desired bioactivity. Method 3. Here I introduced the third dimension by graph neural network processing. So far, we've spoken only about 2D-based design, but the chemical world, as we all know, is not flat. We need to consider stereocenters, for example, the shape of molecules and other 3D relevant properties. So in order to consider to be able to compute three-dimensional conformations, we used a graph-based representation of molecules with vertex and edge features, for example, charges, bond types, element types, and so on, to color this three-dimensional graph. Then, in order to consider local neighborhoods, message passing turned out to be essential when we wish to work with 3D graphs of molecular structures. And then you can use all kinds of, for example, deep neural networks, cascaded, stacked networks, to compute all kinds of properties based on these updated molecular features of the molecular graph. A first example to showcase what's possible with these tools is lead optimization. So we go from the hit to lead stage in de novo design by late stage functionalization. The idea here is to take a molecule and to grow this molecule in some chemically meaningful way so that it can be functionalized without having to synthesize the complete molecule from scratch. In order to do so, we started with a borylation as a very versatile chemical reaction, which is quite fashionable and useful in medicinal chemistry. With our project partners at Roche, we generated training data for this particular graph neural network, and approximately 2,200 reactions were performed. And this is a small data set, but it was sufficient. And these reaction examples were used to train a network, which I will show you on the next slide, to predict potential borylation sites in molecules. We wanted to know from the neural network, will a reaction take place? If yes, where in the molecule and what's the yield? Now here is the schematic of the neural network, deep neural network architecture we used for that task. The input is a molecular graph. Actually, it's a 3D or 2D graph you can use as input. Message passing, multi-head attention-based pooling, and from these two important steps, one can compute these desired outputs. In particular, we added a reaction condition input. So chemists, and that is important, chemists can start to own these deep learning tools by modulating their functionality using well-established chemical knowledge, for example, reaction conditions. The outcome was very surprising. We had extremely good success rates, high success rates for the binary prediction, will a reaction take place, yes or no? If so, where in the molecule reach your selectivity, and the predicted reaction yield. The error of this reaction yield prediction is in the order of the experimental error. Here are a few examples taken from Roche pharma ongoing projects in their pharma discovery projects. On the left-hand side, the input molecule. On the right-hand side, the experimentally observed borylation product. And you will see that in the majority of the cases, the predictions of such a graph neural network were correct. I'd like to highlight one example that is a failure on the lower right. Here, the algorithm predicted that no atom would be a potential substrate or a site of borylation, but in fact we observed borylation. And why is that? Because there was not one example of an aliphatic carbon atom as a substrate for a borylation reaction in the training data. So the system cannot know what it hasn't seen. And this is something also to keep in mind when it comes to training data selection, that we have a rather diverse as possible collection of input data. It's not so much the amount, the number of data points we use for model training, but the diversity of the various instances. Now, method 4, let's put it all together. Let's combine graph-based learning and sequence-based chemical language model to develop a hopefully universal tool for structure-based de novo design, structure-to-sequence relationship learning. And in order to do so, we need to change perspective again when it comes to how we train these deep architectures. Traditionally, we have used a ligand-centric view, ligand-centric chemical space. Each blue dot, a small molecular ligand. We want to predict a new structure, de novo ligand-based de novo design, or predict some activity, for example. But by interactome learning, we train a network, actually a neural network, on the interaction network of proteins and their small molecular ligands. And if we do so, we can now use a graph representation of the binding site as input and a foundation chemical language model as decoder, so that the chemical language model suggests potential ligands for the input ligand binding site, binding pocket. And with that approach, we went into the lab, and this is the very first example we've published on this topic. In fact, starting from PPAR gamma, an empty ligand binding pocket that was not in the training data, this de novo structure generator suggested these two molecules, molecule 1 and 2, they were synthesizable in the lab and had the desired activity and selectivity. Importantly, these two molecules have no anti-target activity in a large anti-target panel. They are not cytochrome P450 substrates up to 10 micromolar, and they are selective towards PPAR gamma. Now, to summarize here, this interactome learning approach implicitly considers ligand selectivity without having to explicitly predict activity. And we have to see as a community whether this approach actually enables ligand design for all protein structures, for AlphaFold predicted protein structures, and that is an ongoing activity. Let me summarize. Rule-based de novo design performs well and reliable when you have only little limited data, scarce data available. Generative AI deep learning models have proven to be robust and deliver novel synthesizable bioactive molecules. The most critical aspect of deep learning in chemistry, in my view, is problem representation, not so much the total amount of data, and not so much the choice of a generative AI, but training data shaping. Most importantly, there is no learning without feedback, and for feedback we need chemistry, because we need to make molecules as a community, synthesize, test them. So as a community, I would say team up chemists with computer scientists, with biologists, and vice versa, because we need much more chemistry. With everything we're doing in this fascinating field area, we should not forget that we cannot escape the edge of chaos, the partial predictability after all, but we can certainly push the envelope together. I thank tons of my co-workers who have contributed to these developments over the past 30 years, my collaborators and sponsors, and you for listening, and I'll be happy to answer any question you may have. Thank you.
Thank you, Gisbert. Thank you for a very structured talk. We are not going to have too much time for questions. Kindly answer what you see on the screen after, but a couple of questions I'd like for you to answer quickly. Given the scarce data problem, and with AlphaFold filling the structured data gap, what type of experimental data are now most critically in need?
In my view, it still is activity data for a broader diversity of targets.
Okay. Off-target effects. So as
Yes.
you design,
So
how do you address this?
Off-targets effect, well, for a long time, we've tried to, and we have developed individual prediction tools for each target, one after the other, one activity prediction after the other, multitask learning, and what have you. So to try to predict explicit bioactivities, either as yes/no decisions, binds, does not bind, or as PKIs, for example. Now, with this interactome learning approach, we try to capture activity and selectivity implicitly rather than explicitly by training the network, the neural network or the deep learning model on the network of interactions, so that only relevant interactions between ligands and targets are considered during the de novo design step.
Thank you, and we'll have to close the session. If you have time, please kindly answer questions in Q&A that we didn't have time for. Thank you very much, Gisbert, for an excellent talk.
Thanks, Alex.